Yes, in the previous video, we had a little bit of discussion about what cellular respiration is and what is the difference between respiration and breathing. And in this video, we're going to go and study in detail about how does the process of respiration occurs. So let's begin. Oops. So as I mentioned before, cellular respiration is a process in which glucose is broken down into, just let me write that down. So you have glucose and you have oxygen. In presence of oxygen, it is broken down into water, carbon dioxide, and energy. So this is basically the equation for the cellular respiration process. And so cellular respiration is basically divided into three processes, which number one is glycolysis, number two is the Krebs cycle, and then number three is electron transport chain. And today our topic of discussion would be about glycolysis. Now, glycolysis is really a simple glycolysis. This is a really simple process and it's really easy. So because what happens is you can just get an idea by just looking at the word, is that glyco, which means it's derived from glucose, which means sugar, and lysis, which means breakdown. It means to break down glucose. So you're basically splitting glucose into halves. So break down. So basically you have six carbon molecule, you break it down into two carbon molecules, each with 3 carbon atoms. And this process occurs within the cytoplasm of the cell. So it occurs within the cytoplasm. Whereas these two processes, these two right here, occur within the mitochondria. And sometimes when you read about mitochondria, you read about it's the powerhouse of the cell where it generates ATP, because, well, this part right here occurs within the cytoplasm and it generates a little bit amount of ATP, whereas these two processes, not so much this, but this process, electron transport chain, which occurs in the cytoplasm, and this also occurs in the cytoplasm, but the electron transport chain makes the huge amount, you can imagine as a bulk of ATP, which is made by the electron transport chain, by this process right here. So but today the topic of our discussion would be about glycolysis. Now let's begin. Basically, when you're studying glycolysis, there's just two things. Like a simple equation can define the whole glycolysis of what glycolysis is. And what it is just, I'm drawing the carbons right here, 3, 4, 5, 6. Whoops, no, this is wrong. So let me remove it. Cut. And this is the right one, actually. Yep, this is the like this. There's another carbon right here, another carbon, and 1 2 3 1 2 3 4 5. So yeah, this is the carbon. This is the carbon. I'm not drawing the hydrogens right here now. So this is a 6-chain carbon molecule, and it goes through a process of 9 steps. So when I'm drawing this arrow right here, it means that it's going through the process, which is divided into 9 steps. 9 different kinds of reaction happens, each with their own enzymes. An enzyme is a protein which helps to speed up or slow down a reaction, or which helps to get the reaction quicker. So it goes through this 9 steps. So this arrow represents 9 steps right here. And as it goes through this process, we get 2 3 carbon two 3-carbon molecules, which are called pyruvates. And as we gain pyruvates, as I said, this whole process helps in making energy. So we get two ADPs plus a phosphate group go into the cell and go into the process, and when they come out as ATP, and you get two of these. And you might be wondering, okay, what is an ATP and what is an ADP? Well, these are I mentioned this in the previous video too, but this is the source of our energy. So cell drives most of its energy from this molecule right here, and to make this molecule is the whole purpose of the cellular respiration of this whole process. So this stands for adenosine triphosphate, which means it has 3 phosphate groups with the adenine group, and this here represents adenosine diphosphate, which means 2 phosphate groups. So when we add another phosphate group to it, it becomes adenosine triphosphate. And so we get 2 molecules of adenosine diphosphate with 2 molecules of phosphate group. They go into this process and come out as ATP. Along with this process, we get NAD plus goes into this process, and as it comes out, it is reduced, which I mentioned in the previous video, plus a hydrogen proton. So we also get 2 molecules of these two. So you might be wondering, okay, ATP is the currency source of energy. What is this? These are also they are rich with energy. These molecules are basically this molecule, the nicotinamide adenine dinucleotide. So these names are really big and sometimes confusing too. But what this molecule is simply it's a coenzyme which carries a lot of energy, and when you go as we go into the electron transport chain, these molecules are responsible for the bulk or for the main source of our ATP. So really important molecule to have around here. So this is pretty much it. Pretty much it. If you are trying to write definition or a short answer on glycolysis, if you just remember this, you're pretty much able to remember the whole process. Now we are going to go a little bit deeper here. So this is basically it. This is the thing which you need to remember if you want to know what glycolysis is. It's a really long process, but this is pretty much it. But now if you want to go a little bit more detail, hopefully I have space. Yeah, I do have space here. Let's do it right here. So basically glycolysis is divided into 2 stages, 2 phases, if I want to be more accurate. One is the investment phase. Investment phase. And no, let me draw it. So this is the investment phase, and other one is payoff phase. Payoff phase. So first we're going to study the investment phase of what an investment phase is. So basically in this phase, as you can get a little bit of idea what this phase is, we are investing. So what are we investing here, Shayan? Tell me more. Well, what we are doing here is we're taking a glucose molecule, 1, 2, 3, 4, 5, 6. I'm drawing it in a straight line because it would be easier for me to explain it. We're taking in a glucose molecule, adding ATP into it. So the investment phase comes here. We're adding ATP into the molecule, and when we apply ATP molecule, it splits into 2, and as it goes through different levels of processes, when we get our pyruvate. So in this phase, we are basically investing ATP. And how do we do that? Well, let's begin. So in this first process, an ATP molecule comes along, goes through this process and leaves out as ADP. So it lost one phosphate group as it entered. And this molecule, 1, 2, 3, 4, 5, and 6. And this molecule became, and this molecule, just a phosphate group is now attached to it right here. So this molecule became glucose 6-phosphate, which basically means like it's a glucose molecule, right? We get that, a 6-carbon molecule of glucose, right? And then the 6th carbon, the 6th right here represents the 6th carbon if we start counting from here to here. And its 6th carbon molecule, we have a phosphate group attached to it, so glucose 6-phosphate. And this is basically this is a nomenclature when you're naming compounds. It's really interesting, so hopefully in the future we'll discuss this too, but let's focus on here today. So we have a glucose 6-phosphate right here, and as we go into the next step, so just to remind you, each of these processes are occurring by their own enzymes, their own specific proteins, which are helping these processes to occur. So now, as we go into after the glucose 6-phosphate, an enzyme comes along, it changes its structure, and in this step, we have the same molecule, 1, 2, 3, 4, 5, and 6, with a phosphate group attached to it, but now we are calling it fructose. We're calling it fructose 6-phosphate. So you might be saying, okay, why are we calling it fructose 6-phosphate? And the reason is that these two molecules may have the same number of carbon, but their structure, their shape is different. So these two are basically called the isomers of each other. So glucose and fructose are the isomers, and isomers is simply a molecule with the same number of same molecular formula but different structural formula. So their both molecular formula is C6H12 and O6. Anyways, let's go. It keeps just getting interesting and interesting. In the next process, in third process, we have another ATP. So in the third process, another ATP comes along and goes out as an ADP. So you might be thinking, okay, another phosphate group attaches to it. Yes, so now this 1, 2, 3, 4, 5, 6. So this is a phosphate group right here, and now we have another phosphate group. This is a bond right here. It's a bond. Let me erase that. Yep, that's a bond right there. So we have two phosphate groups attached to a 6-chain carbon molecule. Now you can figure this out by yourself now, which is quite easy. This is fructose 1,6-diphosphate. It simply means that it's a fructose molecule. Okay, we get that. And in the 1st and 6th carbon, we have two phosphate groups. So that's where the di comes in, the di. And so these molecule is highly rich in energy. So so you might be wondering, it's an unstable molecule right here that we have. And what happens here is that this fructose 1,6-diphosphate splits. It literally breaks into two molecules. Now we're not still in pyruvate. We don't have pyruvate right now, but so we are getting close. We are actually collecting the raw materials for our pyruvates. So these two molecules we get these two molecules in step 4. And what these molecules are called, they are called the G3P. Well, it's easier to call them G3P because what it stands for is glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate. So now these molecules are ready to go in our payoff phase, which we were discussing right here. So these two molecules are ready to go into the payoff phase, which means we are going to get some benefit. So we invested 2 ATP, and now what's the benefit? Well, let's go and let's see what happens. So I'm just going to pick one of these molecules. Let's pick this one, let's pick this one. So in this part, so you're in step 5 right now, right? Yeah, we are in step 5. So in this part, a phosphate group comes along in this process, and at the same time NAD plus is reduced to NADH along with the proton. So we get a proton with ourselves too right here. So when this process occurs, a phosphate group comes along, and so you can imagine where this phosphate group is going to go. 1, 2, 3, and 4. So now we have 1,3 phosphoglycerate. So you don't need to really remember these names, but it's really fun to say these names, so that's why I keep on saying them. 1,3-biphosphoglycerate. So it basically means on the first carbon and the third carbon we have a 2 phosphoglycerates. So now we have this process. Now when this carbon, when our 3 carbon molecule in this form, in this structure or shape, in this form right here, we generate ATP by a process. Now I don't want to confuse you, but it's really interesting. But the process is called substrate level phosphorylation. So this process is a really easy process, but with a really long name. And the process is simply that this 1,3-biphosphoglycerate, this molecule attaches itself to an enzyme. So let me draw an enzyme right here. I'm just drawing a random shape. It's like so this molecule right here, actually pointing my finger at the screen. This molecule right here is attached to this part right here, and there is an ADP group on this part of the molecule. So what happens is when it attaches itself to this enzyme, this phosphate group is released and go to this ADP, and we get an ATP molecule. So from this process, let me scroll down a little bit. I'm running out of space. So as we go into this number 5, right, we're now in number 6 process, an ADP molecule, finally, an ADP molecule comes along and we get an ATP. So now you can imagine, so when one of these phosphate has left with this process right over here, the substrate level phosphorylation, we get a molecule with only one phosphate group attached to it. Now we call it we call it 3-phosphoglycerate. So now in the next process, in the 7th process, this changes its shape and becomes, 2-phosphoglycerate. Now I shouldn't be writing this, just let me write it. Phosphoglycerate. It's a good way to remember these names. So now in the 8th process, water is released from this molecules. Remember that I didn't draw the hydrogens and the carbon and the oxygen because that would take me forever. Oops, and I'm out of space. So just let me do it right here. So in the 8th process, we get a water as a byproduct, and the molecule is now, its shape is changed into what we call, well, in the 8th process, so it's still like this, but its structure is different right now because the bonds are formed in different kinds of ways but still have a 3-carbon molecule, and this is now called PEP. This compound right here is called PEP, and this is a phosphate group, and we call it phosphenol pyruvate. Pyruvate. And after, in the last process, in the 9th process, the pyruvate, another ADP molecule, comes along, leaves out at as ATP, as on the by the process of this substrate phosphorylation. Yep. And we get our 3-carbon chain molecule, which are called pyruvates. Yippee! Finally, pyruvates. And so you might be saying, okay, Shayan, in the starting of this video, you said that we get 2 ATPs, but here I only see basically, here I only see how many ATPs do we have here? Yep, we do have 2 ATPs. Wait a second, where did I do it wrong? We have 2 ADPs and we have 2 phosphate groups. We have 2 ATP. Right. So what did I did wrong here? Let's check. I went all the way through this. I got an ATP. Got an ADP. Hold on, I think I made a little bit of... No, I did everything right, and... Yep, I do get 2 ATPs. Oh yeah, I totally forgot. Sorry about that. I did it right. We do get 2 ADPs. So I actually forgot to mention something. We're not wrong, so don't be afraid that I was saying wrong the whole. I was giving the presentation on this wrong the whole time. No, we are absolutely right. We didn't do anything wrong. The point is, we get 2 ADPs in a sense that, first of all, let's just finish this up. So we get a pyruvate, right? And in this part, so you might be thinking, okay, we get 2, so we have 2 molecules of pyruvate. I just did it for 1, right? So we have 3 glyceraldehyde, 3 phosphate. So we basically get 4 ATPs at the end of this process, one for each part of the 3-carbon molecule. So we do get 3. So we do get 1 for this, 1 for this. So we get 4 ATP at the end of this process. So I almost confused myself. So why do we get 2? Because we invested 2 ATPs right here. So our net amount of ATP, I almost forgot. I actually did forget. The net amount, so we invested 2, we gained 4, so the net profit of our ATP we have here is 2 ATP. So sorry for that inconvenience, and I almost forgot what I was saying when I saw like the equations going wrong, but no, it's not. So let me say that again. So we invest 2 ATP and we get 4. So the net profit, so the net ATP we get is 2. And NAD H's, we also get 2, because for each pyruvate molecule, not pyruvate, the glycer aldehyde 3-phosphate molecule, which is right here. So hope you find that useful, and in the next video we're going to discuss about the Krebs cycle, and after that we're going to study about the electron transport chain. So hope you find that useful. So see you in the next video.